Hello everybody. So today we are going to start another new topic, that is nerve muscle physiology. Under this nerve muscle physiology, the first topic that we are going to understand is nerve and neuron. Okay. So we are starting with the concept of nerve. If you take cross section of any of the peripheral nerve from your body, like for example radial nerve or medial nerve, what you are going to find out, suppose I am taking a cross section. So this is a cross section of a peripheral nerve. Under this cross section, I am going to find out multiple bundles of nerve fiber. So this is one bundle, another bundles of nerve fiber, and another bundles of nerve fiber. These bundles of nerve fiber are known as fascicles, right? So these bundles are known as FASCICLE, fascicles. I told you fascicle means it is bundles of nerve fiber. So what is there inside this fascicle? If I enter inside the fascicle, I'll find out that it contains multiple nerve fiber. This is nothing but axons of a single neuron. So basically nerve fiber means it is the axon of a single neuron. That looks like a fiber. That's why this is known as nerve fiber. So bundles of axons is forming a fascicles. Okay. Multiple fascicle is forming a basically peripheral nerve. So this is again the nerve fiber inside the fascicles. Now you know this nerve fiber, they are surrounded by connective tissue layer. So the connective tissue is covering this individual nerve fiber. So this is another connective tissue. Okay. Similar way, all this nerve fiber will be surrounded by connective tissue. This connective tissue covering is known as endoneurium. Endoneurium, right? Similarly, the whole fascicles will also be surrounded by connective tissue covering. So these are the connective tissue surrounding the fascicles. This is known as perineurium. This is known as perineurium. And the whole nerve fiber, okay, it will also surrounded by some connective tissue covering. So this is loose connective tissue on the surface of the nerve fiber. This loose connective tissue is known as epineurium. Okay, epineurium. You know this thing. Now, the capillaries which supply blood to these neurons or the nerve fiber, they are located in between these fascicles. So these are the capillaries. Okay, these are the capillaries which is supplying blood to this nerve fiber. There is a MCQ here, that which connective tissue layer forms blood neuron barrier. So I am writing here blood neuron barrier. So we have heard about blood brain barrier. So here the question is blood neuron barrier, means whether this endoneurium or this perineurium, which is forming a barrier which is protecting the neuron from blood, right? Answer to this question is nothing but perineurium. Perineurium. So why not endoneurium? Why perineurium? If you look at the perineurium structure, then you will be finding out that perineurium is consist of connective tissue cells. So this connective tissue cells. Now you know that in between this connective tissue cell, the minimum gap which is present, this gap will be bridged by one junction that is known as tight junction. Tight junction. So basically, tight junction we have discussed in general physiology. It is composed of claudine and occludine molecule. It is bridging the gap which is present in the perineurium connective tissue cells. So now this perineurium along with this tight junction forming a continuous barrier, and this barrier is known as blood neuron barrier. So that's why if the question is blood neuron barrier, your answer will be perineurium along with tight junctions form the blood neuron barrier. Now if you look at the research article, they are also saying not only the perineurium, even the endothelium cell of this capillary, so this capillary I am talking about, okay, even the endothelium cell of this capillary, they are also bridged by tight junctions. So the second answer to this question is endothelium cell of this capillary along with tight junctions in between endothelium cell, they are also part of blood neuron barrier. Getting my point? So now with the blood neuron barrier, if we look at the blood brain barrier, if we look at blood brain barrier, then you look at the similarities, right? So blood brain barrier, who forms blood brain barrier? Previously we used to know that astrocytes forms the blood brain barrier, but this concept had been renewed. If you look at the brain capillaries, okay, so this is one of the brain capillary. Obviously it will be lined by endothelium cell, so this is the endothelium cell of brain capillaries. Now I hope all of you know that the capillaries which are present in the brain, they are continuous in nature. What do you mean by continuous capillary? Continuous capillary means the gap which is present in between these two endothelium cells, they are very, very less. In case of brain capillary, remember this gap is less than 2 nanometer, less than 2 nanometers, so very minimum gap. Now this minimum gap is also bridged by again the same kind of bridge, that is nothing but tight junction. So again the tight junction is present in between this endothelium cell. Now if I look at this capillary, I am finding out that endothelium cell along with tight junction, endothelium cell along with tight junction, endothelium cell along with tight junction. It is forming a complete barrier, and this barrier is nothing but your blood brain barrier. So if the question is asked who forms blood brain barrier, your direct answer is structurally it is formed by endothelium cell of brain capillary along with tight junctions, right? So what is the role of astrocyte then? Okay, what is the role of astrocyte then? So this one astrocyte, suppose this is an astrocyte. Okay, so astrocyte extend their processes, and these processes are known as food process of astrocyte. These food process are located at the level of this gap, where the gap is present in between two endothelium cell. But this food process of astrocyte is structurally not part of the blood brain barrier, but astrocyte regulates the permeability of blood brain barrier. It's regulate the permeability of blood brain barrier. So please remember that astrocyte is not structurally part of the blood brain barrier. Blood brain barrier is only formed by the endothelium cell and the tight junction. But what the astrocyte is doing, it is regulating the permeability, means which substances will pass through and which will not. That is determined by astrocyte by secreting certain factor. So this is the similarity between this blood neuron barrier and blood brain barrier. We can find out that everywhere this tight junction is present. Similar to the blood testis barrier. Okay, whenever we will see reproductive physiology, the blood testis barrier is also formed by Sertoli cell along with tight junction. So tight junction is essential for forming a barrier like this blood brain barrier or blood neuron barrier or blood testis barrier, right? So this is all about the basic structural part of a nerve. Now we will be discussing the structure of a single neuron. So next topic is structure of a single neuron. So obviously this is very, very easy because all of you know the structure of a single neuron. So we will only discuss the specific point which are important for our MCQ purpose. Okay, so I am drawing one neuron. Okay, so first this is the cell body part of the neuron. This is the cell body part of the neuron. So these are the dendrites. All of you know dendrites are known as the afferent. Dendrites are known as the afferent part of neuron because all the electrical signal, all the input they will be reaching at the level of dendrites. That's why they are known as the afferent. Okay. Now next part is the cell body or soma. So this is soma. which is nothing but cell body, which is also known as perikaryon. Now the cell body of the neuron, they can be of a variable in size. Okay, it may be small one, it may be large one, depending on the size of the neuron. We have to remember that the smallest size cell body is present in which neuron? Your answer is a granular cell of cerebellum. A granular cell of cerebellum. Okay, it has a size of only 5 micrometer, right? So obvious question is that largest size cell body. Largest size cell body is present in the neurons of anterior horn cell of spinal cord. Anterior horn cell of spinal cord neuron. Their cell body size is approximately 135 micrometer. Very large, largest size cell body of neuron in our body. Right? So these two information is important for MCQ. Now apart from that, we also have to remember certain important organelles inside the cell body or soma. So remember, the cell body or soma contain mitochondria, it contain Golgi body, the normal cell organelle is also present, but we are not very interested in that. Okay, we have to remember cell body contain a special organelle which is known as Nissl bodies or Nissl granules, right? Nissl body or Nissl granule, we know that this is nothing but aggregated rough endoplasmic reticulum. So normal cell contain rough endoplasmic reticulum, that is the protein synthesizing machinery. In case of neuron, all the rough endoplasmic reticulum, they aggregated at a single point, and that is known as Nissl body. So this is rough endoplasmic reticulum and some amount of free ribosomes. Some free ribosomes are also present inside this Nissl body. Nissl body are very rich in RNA material. Because of this RNA material, if you stain the Nissl body, it will be basophilic in nature. It will be basophilic in nature. Means if you stain with Leishman stain or Romanovsky group of stain, it will take the basic dye and it will look basophilic, right? Number 3 is that Nissl body, okay, is never present within axon. Okay, within axon. What I am saying, the Nissl body can be present inside the cytoplasm here. Okay, suppose this is the Nissl body, or it can be present inside the dendrites, also, but it will be never present inside the axoplasm or axonal cytoplasm. It will never present inside the axon. That information is also important. Now, because the Nissl body is a protein synthesizing machinery, the number of Nissl body will be more according to the metabolic activity of the neuron. Means if the neuron is more metabolically active, more number of Nissl body will be there. That's why it is said in book that highest density of Nissl body is found out in motor neuron, which are metabolically very active. So these are the information that may be helpful for MCQ purpose. So please try to remember. Now the second thing is that cell body of neuron also contain certain cell cytoskeleton. Okay, cell cytoskeleton, right? We have discussed the cell cytoskeleton in general physiology chapter. There are microtubule, microfilament and intermediate filament. In case of neuron, the main cytoskeletal organelle is an intermediate filament which is known as neurofilament. So this neurofilament is an intermediate filament which is responsible for maintaining the shell size, neuronal size and shape of a neuron. Okay, that's why you have to remember neurofilament. Apart from that, the normal cell cytoskeleton, like for example microtubules as well as the microfilament, are also present inside the cytoplasm of neuron. Right? Next is that centrioles. Okay, centrioles. What do you think? Centrioles will be present in the neuron or not? We know the function of centriole. They are responsible for mitotic spindle formation. So it is responsible for cell division. But we know that neuron, they do not divide. They remain in the G0 stage of cell cycle. So ideal answer will be that that centrioles are absent in adult neuron, but that is not true. Remember, centrioles are also present in adult neuron also. But the function of this centriole is not for cell division, but it is there to maintain these microtubules, right? So what is the function of this centriole? It maintains microtubules inside the cell cytoplasm. So whenever I am saying that neuron remain in G0 stage, means they do not divide, we have to remember certain exception to this rule. That is presence of neuronal stem cell. Very, very important. Neuronal stem cell. Okay. So I hope you know the meaning of stem cell. Stem cell means they have the property of self renewal, as well as they have the property of differentiation. Means they will maintain the population by self renewal, cell division, as well as they can also differentiate to a different cell population. Neuronal stem cell are present in certain location of the central nervous system. Number 1, hippocampus, right? Particularly the dentate gyrus of hippocampus. This is the area where definite neuronal stem cells are located according to Genome, right? Second area where the neuronal stem cells are located is olfactory bulb region, right? And number 3 CNS neuronal stem cell is subventricular region of lateral ventricles. Subventricular areas of regions of lateral ventricle. Although the neuronal stem cells are rudimentary here, but definite neuronal stem cells are located at the level of hippocampus, followed by olfactory bulb, right? Okay, so we are discussing the different specific point of cell body or soma. So we have discussed centriole and the applied aspect of that. And the last point we have to remember, cell body of the neuron also contains certain pigment in some of the neurons. And because of the pigments, the peculiar color of the neuron will be seen. Like for example, huge amount of melanin pigment, okay, huge amount of melanin pigment are present at the level of substantia nigra neuron, okay, because of which this neuron looks like brown in color, okay. This is also present, melanin is also present at the locus coeruleus nucleus, because of which it looks like bluish in color. Apart from that, you remember huge amount of irons are there at the level of red nucleus, okay, and huge amount of zinc are present at the level of hippocampus. So these are the pigment which are present inside the cell bodies of neuron. So now this is the important point that we have to remember about cell body or soma of the neuron. Now next part of the neuron is the axon part. But remember before that one portion of the cell body, so this is the portion of the cell body where I am marking here. This part here, the cell membrane is a little thicker than other part of the cell body. So this is the membrane is very, very thicker. This is a part of the cell body which is attached with the axon. This part is known as axon hillock. This is known as axon hillock of neuron. So axon hillock is a part of the cell body which remain attached with the axons. And the first part of the axons which starts after this axon hillock, okay, it has a length of 50 to 100 micrometer. Okay, this is known as the initial segment. This is known as the initial segment of neuron. Very, very important. Please remember, don't confuse between axon, hillock and initial segment. They are completely different according to Genome. Okay, so initial segment has very, very important properties. We'll be discussing shortly. But before that, remember the length of this is 50 to 100 micrometer. After that, the long axons will extend up to the level of terminal part of the neuron. So at the terminal part, we all know that axons will be divided into multiple small vesicular region, which are nothing but axon knob. These are known as axon knob or telodendria. Axon knob or telodendria. And we all know that this telodendria is the region where the vesicles will be present, and inside this vesicle various neurotransmitter will be present according to the type of neuron. It may be acetylcholine, it may be glutamin, whatever. But these are the vesicles which contain neurotransmitter containing vesicles. Okay. Now, apart from all this, we know that if the neuron is a myelinated neuron, so myelin sheath will also be present. So this is the myelin sheath, right? This is the myelin sheath. Okay. This is another myelin sheath. Okay, another myelin sheath. Now there is an MCQ. What is the length of a myelin sheath? So they are asking what is the length of this. You remember that in long neuron, the myelin sheath can be of up to 1 millimeter in length. Single myelin sheath can be of 1 millimeter in length. And we also know that in between two myelin sheath, the region is very, very popular for MCQ. So in between these two myelin sheath, this region is 1 micrometer in length, and this is nothing but known as nodes of Ranvier. This is known as nodes of Ranvier, right? So these are all the structural part of a neuron that you have to remember about your MCQ, right? Now after this structural part, we are going to discuss the functional areas of neuron, different functional areas of neuron. Okay, so before functional areas of neuron, I'm going to give you some data, okay, like the neuronal number and all. So if the question is, what is the total number of neurons, present in the central nervous system, your answer is 100 billions, right? That is 10 to the power 11. Then second question, number of glial cell in the central nervous system is even more than neuron, and it is said that glial cell number are 2 to 10 times more, 2 to 10 times more than neuronal number. The third thing is that in CNS, on an average, one neuron can form up to 2000 synapses. 2000 synapses. Getting my point? So we all know that this axon knob, all of these individual axon knob, they can form a single synapse. So in these cases, in this diagram, I can find out that 3 axon knobs are there. So this neuron will form 3 synapses. But that is not the actual number. Actually, one neuron in the central nervous system can form 2000 synapses. So your total number of neuron is 10 to the power 11, and one neuron is forming 2000 synapses. So if the question is total number of synapses in the central nervous system, then your answer is 2 multiplied by 10 to the power 14. Very simple. So now you look at the numbers are very, very huge. The neuron, the glial cells, synapses, these are very complex areas. CNS is a very complex areas. That's why there is a very valid question in the NEET entrance examination, that how many percentage of human genome is responsible for CNS development. Oh my God. So look at, so CNS development, I can understand that it will be a complex process, obviously. They are asking that CNS development is based on how much percentage of human genome or human genes. Your answer is 40% of the total human genomes is responsible for CNS development. So question, how many total numbers of genes are there in human genome? Okay, recent concept is that between 19,000 to 22,000, this is the total number of human genes, human genome. Okay, but if you want to know the exact number, that is 21,306, this is the exact number of human gene at present according to the recent literature. Although different literature are there and they are varied in number, but this is one of the literature is saying approximate 21,306 is the total number of human genes in genome, right? So this is certain database MCQ. Okay, now we are coming to the functional areas of neuron. So functional areas of neuron. Your MCQ will be like this. Number 1, graded electrogenesis. Graded electrogenesis occurs in which part of the neuron? This is a little controversial question. Some of the MCQ book has given wrong answer. Remember, the answer to this question, graded electrogenesis, is soma and dendrites. So, dendrites and soma, cell body. Okay. What is graded electrogenesis? You will understand this in the next class. Okay. But for the timing, you remember, graded electrogenesis means it is a kind of local potential development on neuron. Okay. So whenever you stimulate a neuron, it will produce a local potential, graded potential, and that graded potential will originate at the level of dendrites and cell body. But dendrites and cell body, they cannot produce action potential. So the second question is action potential initiations occurs in which part of the neuron? Action potential initiation, very valid question, because you are producing a local potential in the dendrites and cell body, but they cannot produce action potential. So where the action potential will start for the first time? To understand this concept, you have to answer another question, that is voltage gated sodium channel, where it is maximally present. Why voltage gated sodium channel? Because we all know that action potential in the neuron is based on a channel, and that is voltage gated sodium channel. Voltage gated sodium channel. So the area where the voltage gated sodium channel is maximum, that area will have a minimum threshold, least threshold, and the action potential will be initiated in that area only. That is the concept. More number of sodium channel, low is the threshold, and maximum sodium channel, lowest is the threshold. Lowest threshold means that is the area where action potential will be initiated. So if you could answer this question first, voltage gated sodium channel, where it is present maximally, the answer is node of Ranvier, whether it is sensory neuron or motor neuron. Genong is saying that at the level of node of Ranvier, voltage gated sodium channel number is around 2000 to 12,000 per square micrometer area. Very high. If this is not given in the choices, then the second choice is initial segment of neuron. Initial segment of neuron, where number of voltage gated sodium channel is from 350 to 500 per square micrometer area, right? But if you look at the cell body or dendrites, means soma or dendrites, number of voltage gated sodium channel are very less there, and that's why I told you earlier that they cannot produce action potential. Similarly, if you look at the telodendria, it also contains very low number of voltage gated sodium channel, and the question is least number of voltage gated sodium channel is present on the surface of myelin sheath. On surface of myelin sheath, right? So that is the area where voltage gated sodium channel are minimum. That means if you look at this neuron, voltage gated sodium channel is maximum in this region, second maximum in this initial segment, and the least is on the surface of this myelin sheath. This is 3 important MCQ that you must have to remember, right? Very, very important. Now, so if we look at the question that action potential initiation will occur in which region of the nephron, remember the answer should be node of Ranvier, but there is a little problem here. If the question is action potential initiation in case of sensory neuron, then your definite answer is node of Ranvier. But if the question is about motor neuron or any other type of interneuron, then your answer will be initial segment. So what I told, please note down that if the question is in case of motor neuron where the action potential is initiated, in case of motor neuron where the action potential is initiated, right, your answer is initial segment. But if the question is in case of sensory neuron where the action potential is initiated or started for the first time, your answer is node of Ranvier, right? For motor neuron or interneuron, it is initial segment. For sensory neuron, it is node of Ranvier. Just put this information in your brain. Now, if you want to understand why this difference is there, sir, why this difference? Okay, I can recall, but why this difference is there? Then you look at the picture. Okay, so this is the spinal cord. Okay, cross section of the spinal cord. This is central canal. You know that the motor neuron, they are located in the anterior region of the spinal cord. So this is my motor neuron, and this is the anterior part of spinal cord, and this is the posterior dorsal. And you know also that the sensory neuron, they enter into the spinal cord through the dorsal root region. So this is the sensory neuron, okay, and the cell body is here. This is the dorsal root ganglia where the cell body is located. I hope all of you know that this is my sensory neuron, right? Now, if the sensory neuron is a myelinated neuron, so these are the myelin sheath. These are the myelin sheath. Okay. Now, remember 2 things. For action potential initiation, you require huge amount of voltage gated sodium channel. And second thing is that action potential will be initiated in an area which is close to stimulus point. What I told? If you give a stimulus here, action potential will try to originate in an area near to this where the voltage gated sodium channel is high. It is not that if you stimulate here, action potential will be originated in a distance area. No, that is not possible. Okay, so this is sensory neuron, and I can imagine that if this is my skin, okay, so I am giving a stimulus at the surface of the skin. Suppose this is a kind of touch stimulus. Okay, in presence of stimulus, the touch receptor will be activated here. Whenever the touch receptor is activated here, this is the stimulus point. Action potential will try to originate in an area which is near to this stimulus point, which is near to this receptor, and which contain huge amount of voltage gated sodium channel. So it will search this area. So it is searching. Whenever it is reaching at the level of this node of Ranvier, it is getting huge number of voltage gated sodium channel, and that's why the action potential will be initiated in the first node of Ranvier here, and then it will jump from one node to other node, and it will reach up to the level of spinal cord. This type of conduction is known as saltatory conduction of action potential. We all know. So in case of sensory neuron, you are giving stimulation at the level of receptor, and close to the receptor, the first node of Ranvier is located, and action potential will be initiated there. Right? Got it? Now look at the motor neuron. Okay, who gives stimulation to the motor neuron? Motor neuron gets their stimulation from cerebral cortex. So brain is giving stimulation to motor neuron. Where the stimulation is going on? The stimulation is going on at the level of dendrites or at the level of cell body, because synapse is formed there. So brain is stimulating the motor neuron at the level of dendrites and cell body. Can dendrites and cell body produce action potential? Answer no. Why? Because voltage gated sodium channel is very low there. So now this stimulus point will search an area where voltage gated sodium channel is maximum, and whenever it is reaching at the level of this initial segment, this will get huge number of voltage gated sodium channel, and action potential will be initiated at the initial segment region, and then the action potential will jump from one node to another node. So that's why in case of motor neuron, the action potential is initiated at the level of initial segment, okay, because that is close to the stimulus point. In case of sensory neuron, the action potential is initiated in the node of Ranvier because that is close to the receptor, that is close to the stimulus point. So please remember these functional areas of the neuron. These are very important for physiology MCQ. Okay. Now we are going to discuss the different types of neuron. Okay. So different types of neuron. Okay. Just now we have discussed the sensory neuron. Okay. And we have discussed the motor neuron. So if we look at this neuron, okay, what type of neuron they are? Okay, again this is a very simple topic. You know that there are different types of neuron. One of the neuron is known as the unipolar neuron. Unipolar neuron means very simple, one cell body, and from the cell body one single process. This is unipolar neuron. According to Genome, unipolar neuron is not present in our body. Unipolar neuron is present only in invertebrates. But in anatomy book, it is given the examples of unipolar neuron in our body is mesencephalic nucleus of fifth cranial nerve. Mesencephalic nucleus of fifth cranial nerve. So try to remember this point. Okay. Second is that pseudo unipolar neuron, right? So it is a pseudo and unipolar. Okay. So this is cell body. From cell body, one process is arising, and that process is dividing into two. Okay. So from the cell body, only one process, and that is dividing into two. That's why this is known as pseudo unipolar neuron. All the sensory neuron are example of pseudo unipolar neuron. Just now we have discussed here, just now. So this is a sensory neuron. Okay, if you look at this, okay, this is a cell body, one process, and that is dividing into two. So this is known as pseudo unipolar neuron. I told you the cell body of the sensory neuron are located at the level of dorsal root ganglia. That's why the sensory neuron are also known as dorsal root ganglion neuron. Dorsal root ganglia neuron. Right? So this is pseudo-unipolar. Next variety is very simple, that is the bipolar neuron. Cell body, from cell body, two branches. Both of them are attached with the cell body. That's why this is known as bipolar neuron. We all know bipolar neurons are located at the level of retina. It is also located at the level of olfactory epithelium. The olfactory sensory neuron, okay, which are located at the roof of our nose, those olfactory sensory neuron are also example of bipolar neuron. And lastly, the multipolar neuron. The motor neuron that we have discussed just now. Cell body, from the cell body, multiple processes which are nothing but dendrites, and a single large process which is nothing but axons. This is typical of multipolar neuron. So examples are many, but the typical example is motor neuron. They are multipolar neuron. So this is all about the basic structural functional aspect of neuron. Now one topic we will discuss at the end of this chapter, that is axonal transport. What I told, axonal transport. But first you look at the logic. Okay. So we have discussed the Nissl granules or Nissl body. They are the aggregation of rough endoplasmic reticulum. Where they are located? Inside the cell bodies or dendrites. So suppose this Nissl body is producing a protein molecule. Okay, so this is a protein molecule. So this protein may be channels, maybe pump, maybe sodium, potassium, ATP, whatever. Now I have to transport this protein from the level of this cell body up to the level of this axon end. From cell body to axon end. how to transport this protein inside the cell, and that is nothing but axonal transport or intracellular transport within the cell. This process is similar in every cell, okay, but generally this is well known or well studied in case of neuron. So in case of neuron, the transport within the cytoplasm of the neuron is known as axonal transport. Okay, so how the axonal transport occurs. Okay, so I am drawing a diagram. Diagram is important. Okay, so this is a neuron, although the shape of the neuron is not typical. Okay, anyway, so this is a neuron. Okay, and this is the cell body. So suppose this is my rough endoplasmic reticulum, RER, rough endoplasmic reticulum, and it is producing one protein. Okay, first of all, this protein will be packed inside certain vesicles. So this is vesicles. Inside this vesicle, the protein is packed, and this packaging will be done by Golgi body. We know that thing. Now I have to transport this vesicle from cell body to axonal end. What are there inside this axon? There are certain cell cytoskeleton, and one of such important cytoskeleton which is present inside this is microtubules. So I am drawing the microtubules. So this is my microtubules. Okay, in general physiology we have discussed that microtubule is a dynamic cell cytoskeleton which has a growing end which is known as plus end and a non-growing end which is known as minus end. So plus end will be directed in this direction. Why? Because in case of axon, the growing end is towards the axonal end. Okay, in case of neuron, the growing end is towards the axonal end. So this axonal end means that is the growing end. So microtubule plus end will also be directed in that direction, and the other end of the microtubule is known as a negative end or minus end. Right. Remember that this microtubule is a pathway for the transport of this vesicle. Okay, I have to transport this vesicle, right? So this vesicle, how it will be transported? It will be transported on a pathway, and that pathway is microtubule. Now on this pathway, there are some motor system. So this is one motor system which is running on this pathway. Okay, which is running on this pathway. You imagine the Indian railway system. Okay, so one train is moving on the railway path. Okay, and this train is moving from negative end towards the positive end. So this is a motor. Okay, in molecular biology terms, this is a molecular motor. So I am giving this name. This is nothing but molecular motor. The name of this molecular motor is kinesin. Kinesin. So what we are going to understand? We are going to understand axonal transport. Just now we have discussed that axonal transport is based on certain molecular motor molecules, and the first molecular motor that we have just told is nothing but kinesin. If I ask you kinesin is moving from which to which direction, your answer will be kinesin is a plus end directed motor. Plus end directed motor. Plus end directed motor means it is moving from minus towards the plus end. This plus end directed motor is this kind of transport is also known as anterograde transport. Anterograde transport. So kinesin is an anterograde transport. So how this vesicle will be transported? Now you look at the whole process. First of all, this vesicle will be taken up by this kinesin molecular motor, and then the kinesin molecular motor will move onto this microtubule, okay, like the Indian railway system, okay, and it will move until and unless is reached at the end of the microtubule. At the end of this microtubule, it will release the vesicles within which the protein is already present, right? So by this way, any vesicles, any cell organelle which are moving from cell body to axon end, it takes help of microtubule and it takes help of kinesin molecular motor. Now question is, if some substances is moving in the opposite direction, sir, suppose one virus has already gained entry into this region, means at the region of the synapse, one virus has entered into the neuron. So how this virus will move from this axon end towards the cell body, means now I have to transport this virus in the opposite direction, or maybe certain growth factor, or maybe certain toxins. We know that tetanus toxin, it moves from periphery towards the central nervous system, means in a retrograde direction. So how this tetanus toxin or how this virus will move from axonal end to cell body? Let's discuss. So another Indian railway system is here. Okay. So this railway is moving in the opposite direction. Okay, it is moving in the opposite direction, and the name of this molecular motor or this railway system is nothing but dynein, dynein, which always moves in the retrograde direction. So second molecular motor that I am writing here is dynein, 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 and dynein is a negative end directed molecular motor. Negative end directed molecular motor. Negative end directed means I can also say that this is the retrograde type of transport. Now pathway for both of this kinesin and dynein, we have already discussed that both of them work on microtubules. Why I am using the word work on? Because if you look at the actual movement of this molecule, okay, kinesin and dynein, they work like this. They just work on the surface of this microtubule like this. There are videos in YouTube, so you can look at that. Kinesin and dynein, they have taken images of this movement. Okay, and just like we are working on you are working on pathway, similarly the kinesin and dynein, they also work on the surface of this microtubule. That's why for both kinesin and dynein molecular motor, pathway is the microtubule, but one of them is the anterograde transport, that is the kinesin, and one of them is dedicated for retrograde transport, that is dynein. Okay, but you must have noted that I already told that kinesin will bring the vesicle up to the end of the microtubule. But question is, how this vesicle will move from end of the microtubule up to the region of the cell membrane? There is again a long distance from the end of the microtubule up to the level of cell membrane. Okay, so I told you Indian railway system. You know that Indian railway system means there is an Rajdhani Express and there are multiple local trains. So this kinesin and dyneins, they are like Rajdhani Express, but you also know there are local trains. So there are pathways for local train which extend from the end of microtubule up to the cell membrane, and this pathway is formed by microfilament. I hope all of you remember microfilament means they are the actin molecule. So actin is the pathway for local train. So for local train, who is the molecular motor? The molecular motor for this local train is myosin molecular motor. They can move in both the direction. So this is the molecular motor which is nothing but myosin. Myosin can work on this pathway, and this pathway means that is the actin pathway, that is the microfilament pathway. On this microfilament pathway, the molecular motor which travel is nothing but myosin molecular motor, which is a bidirectional motor. So you may be a little bit confused. Myosin means that myosin, sir, which is responsible for muscle contraction? Answer is yes. Answer is yes. But obviously there is a different sub-variety of myosin. What do you mean by that? Till date, you know, there are 18 different sub-varieties of myosin has been discovered. We do not know the function of all of this variety, but we know that muscle contraction, okay, muscle contraction is based on myosin type 2. So please note down, myosin type 2 is responsible for muscle contraction. Any kind of muscle contraction, skeletal muscle, cardiac muscle, smooth muscle, all myosin type 2. But the myosin which is acting as a molecular motor, okay, so molecular motor myosin. So third is the myosin molecular motor, okay. They are bidirectional molecular motor. They work on microfilament. Okay, which subtype of myosin? Myosin type 5, okay, myosin type 1, myosin type 6. Why I am writing 5 first? Because that is the most important myosin which is acting as a molecular motor, and second most important is type 1. Myosin type 5 and myosin type 1, these are the myosin which are generally positive end directed molecular motor myosin, and myosin type 6, this is basically negative end directed myosin molecular motor. So basically I told you myosin is a bidirectional molecular motor. That does not mean all are bidirectional. Some of the myosins are positive end directed and some of them are negative end directed. Even if you don't remember this sub variety, you just remember myosin is a bidirectional molecular motor. But okay, myosin work on microfilament, and kinesin and dynein, they work on microtubules. So this is about the axonal transport, how it is happening, okay, with the help of certain molecular motor. Depending on the velocity of this transport, depending on the speed of this transport, axonal transport is divided into 3 subvariety. So depending on the velocity of transport, number 1, okay, so types of axonal transport. Number 1, fast anterograde, okay. Number 2, fast retrograde, okay. And number 3, slow andrograde. There is no slow retrograde. Slow andrograde is there, but retrograde is not there. So this is the 3 different types of axonal transport depending on the speed or velocity. So first one, fast anterograde, obviously the molecular motor is kinesin, okay, and the speed or velocity is 400 millimeter per day. 400 millimeter per day, right? The fast retrograde, the molecular motor is dynein and speed is 200 millimeter per day. Third one, molecular motor is not known, okay, but the speed of this is 0.5 to 10 millimeter per day, right? So slow anterograde, the molecular motor is still not known. Now, what are the molecule that is transported by fast anterograde transport? Okay, I told you already, any kind of vesicles which are derived from Golgi body. So Golgi derived vesicles will be transported by fast anterograde transport. Second, any kind of cell organelle, like for example mitochondria. Suppose you want to transport mitochondria from cell body to axonal end. Again, you require the help of kinesin. The speed is 400 millimeter per day. And retrograde, I told you already, there are certain viruses, okay, like for example herpes simplex virus. There are certain toxins, like for example tetanus toxins, and there are certain growth factors secreted by different tissues. So growth factors, they are transported in retrograde direction by dynein. And for slow anterograde transport, the molecule which are transported by the slow anterograde transport are component of microtubules and components of microfilament. We have discussed in general physiology that microtubule is composed of tubulin molecules. So if you want to increase the growth of the microtubule, you require the subunit which are nothing but tubulins, and those tubulin subunit will be transported by the slow anterograde transport. The subunit of microfilament, that is the G-actin, they are also transported by slow anterograde type of transport. So this is all about axonal transport that you have to remember for your MCQ. Please remember, this is a topic which is given in Genom also, and the question has been asked in INI set type of examination. So this is all about the first chapter. Thank you.